Hello, I'm Dr. Payal Dande. Welcome to the learning episode of B.Pharm on a successive unit of glycoside part 3 under the section of Chemistry of Natural Products in semester 3 of B.Pharm. Before we start, let's first have a look onto our contents and the learning objectives. To start with, in module 1, we will have a brief introduction of the saponin glycoside, whereas in module 2, we are going to see the classification of saponin glycoside based upon its different chemical structure. In module 3, we are going to discuss about their extraction techniques, the identification test, and their therapeutic significance. In module 4, we are going to discuss about the flavonoid glycosides. Module 5, we are going to look upon the isothiocyanate glycoside by giving an extra importance to cinegrine as an example. In module 6, we are going to discuss about the coumarin glycoside, and in our last module, we are going to talk about the bitter glycosides. In our last lecture of glycosides part 2, we discussed about the two important types of glycoside. Remember, they were cardiac glycoside and anthraquinone glycoside. We discussed about their chemistry, their structure-activity relationship, the drug source, the chemical test used for their identification, and also about their therapeutic significance. In continuation to the same, now let's discuss and understand the chemistry and pharmacological significance of the various other important glycosides of our modules. Remember, we said in our last lecture that with the change in the glycon moiety in glycosides, there is a vast alteration seen in the therapeutic effect they exert. This becomes much more evident when we talk about the saponin glycosides. So here we are to start with saponin glycosides. Saponin glycosides are a group of natural occurring compounds that are amorphous and colloidal in nature. Now they are widely distributed in the higher plants and are being used for their medicinal as well as commercial significance. Let's look a little to the history of saponin glycoside. They have derived their name from a plant known as soapwood, from the genus Saponaria, because they form soap. They have the ability of forming soap. That's why sapo. Sapo in Latin means soap. Yes, these glycosides have the ability to form lasting foam when shaken in aqueous solution. In fact, earlier they were used as detergents to replace the soaps. Can any one of you guess a plant fruit that is still being utilized in hair care for making herbal shampoos at home in India? Well, it's called as Rita in Hindi and famously known as soap nut or wash nut, and the plant bearing this fruit is known as Sapindis mucrosi. Besides their foaming property, they were also used as pesticides to stun the fish in the olden times, as most of the saponins have hemolytic properties and they are toxic to cold-blooded animals, specifically to fish. You see, plants containing saponin compounds were used as food poison to catch the fish in many countries. However, exceptionally, we also have licorice, which is called as mulethi or jeshtman in India, that contains saponin glycoside and is used as food as a sweetening agent. Okay, see the variety and see the differences. Now let's move forward to know more about its sources, chemistry, and the physical properties along with its medicinal significance. As we said, historically saponins were known to be found only in higher plants, but of late they are also being found to be present in marine organisms. The important plant families that contain saponin glycosides are Araliaceae, Aceraceae, Asclepidaceae, Berberidaceae, Cucurbitaceae, Caryophyllaceae, Compositae, Dioscoraceae, Hippocastanaceae, Leguminosae, Rubiaceae, Solanaceae, Sapindaceae, and so on. They are also found in monocot families such as Liliaceae, Dioscoraceae, and Amaryllidaceae. They are also located in various parts of the plant such as roots, rhizomes, stolons, tubers, stems, leaves, flowers, seeds, as well as fruits. Some of the important plant sources containing saponin glycosides are given on your screen. Few of the saponin glycosides with their drugs, their biological sources as well as uses. Let's first start with the asiaticosides, which are found in dried herbs of Centralia asiatica coming from the family Umbelliferae. They are commonly known as Jal Brahmi and they are used as nervine tonic. Similarly, we also have bacoside A and B, which comes from the dried leaves and stems of Bacopa monieri from the family Scrophulariaceae. They are commonly known as Bacopa or Brahmi and they are also used as nervine tonic. Then we have the most common charantine and momorodicine coming from our common fruit known as Karela or bitter gourd. These are dried fruits of Momordica charantia from the family Cucurbitaceae and we all know that they are being used as a hypoglycemic agent. Then we have another famous compound known as diosgenin, which is a steroidal sapogenin. These are found in the dried tubers of Dioscorea deltoida from the family Dioscoreaceae, which is commonly known as yam or Dioscorea, and they were the precursors for the synthesis of various steroidal hormones. Once again, we have glycyrrhizine, which is a triterpenoid saponin coming from the roots and stolons of Glycyrrhiza glabra, commonly known as licorice, jeshmad or muletti from the family Leguminosae. They are being utilized as expectorant, anti-ulcer and even hypoglycemic agents. Another we have ginsenoside and panaxoside, which comes from the dried roots of Panax ginseng from the family Araliaceae, and they are commonly known as ginseng. They are being used as an adaptogen, tonic and even as a stimulant drug. We have the most famous drug called as Shatavrine 1 and 2 coming from the dried roots and leaves of Asparagus racemosus of the family Liliaceae, which is famously used as a galactagogue. Similarly, we also have senegine, which is a polygalic acid, a triterpenoid saponin coming from the roots of Polygala senega from the family Polygalaceae, and they are being utilized as stimulant and expectorant. So here we are with the various saponin glycoside either in the form of triterpenoids or in the form of steroids as well as in the form of sapotoxins. Another important fruit known as gokru or tribulus gives pterosterosine A and E along with tribulosine, which are steroidal saponins which are found in the fruits of Tribulus terrestris from the family Zygophyllaceae and is being used as diuretic drug. Now let's look upon the physiochemical properties of saponin. Saponin glycoside consists of hydrophilic glycoside moieties attached to a lipophilic triterpenoid or steroidal nucleus. They are generally colorless, white or milky white, amorphous powder that are difficult to crystallize. They are optical active agents with steroidal saponins and sapogenins being levorotatory in nature. The unsaturated or saturated acyl derivatives normally show smaller optical rotation value than their corresponding saturated saponins. They have high melting points ranging around from 200 to 350 degree and generally decompose before melting. They form colloidal solutions with water and they are soluble in alcohol too, in even dilute alcohol, while insoluble in all the organic non-polar solvents such as ether, benzene, chloroform and lipophilic organic solvents. They are excellent emulsifying agent, that is, they can help in modifying the surface tension. They are surface active agents and produces foam whenever they are dissolved in water. Saponins have hemolytic properties too. They precipitate the cholesterol and lecithins that exist in the membrane of the red blood cells and thus hemoglobin is liberated. Thus they are extremely toxic when injected into the bloodstream. However, they are not harmful when taken orally. According to the nature, they are classified as acidic saponin, neutral saponin and alkaline saponins. Now let's look upon to the classification of saponin glycoside which is based on their chemical structure. Have you noticed in the above table where some plants were containing triterpenoid saponins, the others showed the presence of steroidal sapogenins, while in some they were also containing sapotoxins. Do you remember the definition of glycoside learned at the introductory chapter of this topic? Yes, we said that the glycosides are organic compounds which on hydrolysis yield sugar moiety and a non-sugar moiety. The sugar moiety is called as glycon and the non-sugar moiety is either known as glycon or genin. Similarly, the glycon moiety of saponin glycosides are called as sapogenin, while some harmful sapogenin are even termed as sapotoxins. Well, based on the structure of glycon, they can be classified into two major types, the triterpenoid saponin glycoside and the steroidal saponin glycosides. Both the type of glycon is attached to different sugar moieties and uronic acids. Well, a third type is also mentioned in some texts as steroidal amine or steroidal alkaloids as they contain nitrogen atom. However, they are considered under pseudoalkaloids. So let's now discuss the difference between these two types of glycosides and also evaluate if this difference in the chemical structure affect their pharmacological activity. Let's start first with the triterpenoid saponin glycoside. Talking first about the triterpenoid saponin glycoside, these group contains pentacyclic nucleus with 30 carbon atoms linked to a sugar moiety at C3 carbon atom. They arise from the cyclization of 3s-2,3 dihydrosqualene. They are structurally diverse compounds containing 4 to 5 basic ring systems in the nucleus. Based on the structure, they are further classified into three groups as alpha-amarine, which is also known as ursane, beta-amarine, which is also known as oleonane, and lupiol, which is known as lupine. They majorly belong to this oleonane, ursane and lupane type, but sometimes they are also seen as hopane, germinicane or damarane type. The hydroxyl group is usually located at C3 position. They are majorly found to be acidic saponin as they contain carboxyl group. Triterpenoidal saponins also differ in their distribution pattern from steroidal glycon. They are mostly found in the dicot families like Berberidaceae, Caryophyllaceae, Chenopodiaceae, Compositae, Polygalaceae, to name a few. Some are even found in marine animals and some also are found in pteridophytas. Now let's look upon the structure elements that are characterizing this series of triterpenoidal saponin glycosides. There has to be a common occurrence of double bond between C12 and C13 carbon atom. An oxidation also is being seen at C23, C28 and C30 methyl group, where forming the hydroxymethyl group, aldehyde or carboxyl moiety. The oxidation at 2, 7, 11, 15, 16, 21 and 22 hydroxyl group, especially at C11 to form the ketone. Sometimes the glycon is partially esterified by low molecular weight aliphatic acids. Example, thiasaponins or gymnamic acid as seen in Gymnema species. Similarly, we also have acene in horse chestnut seeds. Now let's talk about the second type of saponin glycoside, which is known as steroidal saponin glycoside. These are the saponin glycosides which contain 27 carbon atom sapogenin nucleus and includes either a 6-ring spirostane or a 5-ring furostane skeleton. If the hydroxyl group at C26 is engaged in glycosidic linkage, then the structure remains pentacyclic and it's known as furostane. Spirostane glycosides are found majorly in bulbs, roots and seeds. They are generally neutral saponins as they do not contain carboxyl groups. Steroidal saponins are less widely distributed in nature than triterpenoidal saponin glycosides. They are mainly found in the monocot families such as Liliaceae, Discoriaceae and Agavaceae, while Some of them are also found in dicot families such as Leguminosae, Scrophulariaceae. Some of them are also found in Amaryllidaceae or Bromeliaceae. Smilax and Dioscorias are the most common and famous genera containing steroidal saponin glycosides. Now let's look upon to the structural element that characterize this series. First, it contains either hexacyclic spirostane or pentacyclic furostane skeleton. The presence of hydroxyl group at C3 position is must. Sugar chain is usually connected from C3 position. A hydroxyl group may even be present at carbon 1, carbon 2, carbon 5 and C6, whereas they are less commonly found at C17 and C24 as seen only in Liliaceae family. The example is chlorogenin. In some cases, lactonization is seen forming a spirostane 26-one. Example is solanum, where the demethylation or sulfation of hydroxyl group occurs. Spirostane structure may have both S or R configuration at C25 carbon atom, giving rise to two series, one neosapogenin or spirostenols. As they possess the steroidal nucleus, they are used as a precursor for the synthesis of steroidal hormones like testosterone, progesterone and estradiol, even for other steroidal compounds like vitamin D, cardiac glycosides and corticoids like betamethasone and cortisone acetate. Now let's look upon the classification which is based on glycosidic chain. Based on the presence of number of glycosides in side chain, they are called as monodesmoside, which contains one glycosidic group, or saccharide moiety at C3 position. Or either they can even be called as bisdesmosides, which contains two glycosidic group or saccharide moiety, one at C3 position and another at C28 position. The common sugar moiety seen attached to sapogenin are usually D-glucose, D-galactose, L-arabinose, L-raminose, D-xylose, D-fucose, D-glucouronic acid. Although amino sugars are present only in exceptional cases, they are usually present as one or two linear or branch oligosaccharides that are linked to a glycan by either an ether or an ester bond. Now let's look upon their extraction, identification test and their pharmacological significance. As saponin glycosides are polar compounds, they are soluble in water and alcohol. The crude drug powder may be first extracted with a non-polar solvent such as petroleum ether to separate the fatty acids and other unsaponifiable matters, and then it can be extracted using either alcohol or water. Further separation and purification of saponin glycoside is a little delicate and difficult procedure as they are highly polar, relatively fragile and closely linked to each other and found as a complex mixture. Now let's see the chemical tests which are used for the identification of saponin glycoside. The most simplest and the easy test to identify the presence of saponin glycoside in crude drugs is foaming test. Formation of a stable and a persistent foam with water signifies the presence of saponin in the drug. As these glycosides contain triterpene and steroid glycan, it can also be detected by Lieberman-Burcher test and Salkowski reactions. As some saponins have the ability to hemolyze red blood cells, hemolytic test is also used for their identification. Now let's look upon to the various procedures of this test. Coming first to the foaming test. You have to take the drug extract or drug powder with water and shake vigorously for 30 seconds. Then allow it to stand. Formation of a stable persistent foam will indicate the presence of saponin glycoside in the drug sample or the extract. Similarly, let's look upon the hemolytic test. The drug extract or the drug powder is added to a drop of blood sample on glass. The presence of a hemolytic zone will confirm the presence of saponin glycoside in the drug. Now let's look for Lieberman-Burcher test. This test helps us to differentiate between triterpenoid and steroidal moiety. For this test, First, we have to dissolve little quantity of drug compound in chloroform and then add equal volume of acetic anhydride to it. Add 1 or 2 drops of concentrated sulfuric acid from the side of the inclined test tube. Pink to red color shows the presence of triterpenic glycon, while the bluish green color will indicate the presence of steroidal glycon in the saponin compound. We also have Salkowski reaction, which will confirm the presence of steroidal moiety. When concentrated sulfuric acid is added to a solution of drug compound in chloroform solution, a red color is produced in the chloroform layer, which confirms the presence of a steroidal glycon. Having known about the different identification tests which we can use for the saponin glycoside, let's now move forward to see their pharmacological activities. Saponins are found to have significant diversity in their structure, and these structural diversity leads to varied biological activities. In fact, they are found to exert structure-related biological activities. The presence of different basic ring structure, different substitution such as hydroxyl, hydroxymethyls, carboxyls and acyl groups on the basic nucleus, the difference in their composition or even the arrangements can actually make a change in their pharmacological activities. Not only this, but the difference in the number of sugar chains attached can also make a difference in their pharmacological activity. All the above differences accounts for substantial structural diversity of these saponins and thus leads to different action. Based on the above structural diversity, they exert several pharmacological activities, from being anti-inflammatory, anti-ulcer, antimicrobial, hemolytic, and cancer-related agents. They are also known to be immunomodulating agents, mollicidal in action, spermicidal in action, and also gives abortifacient activities. They are also known to exert their action on the cardiovascular, central nervous system, and endocrine systems. Recently, they are used as an adjuvant for vaccine as immunostimulatory complexes and also for the enhancement of synergistic effect of immunotoxins. Some of the most common pharmacological uses of saponins are steroidal sapogenins such as diosgenin, isolated from the rhizome of Dioscorea species, are used as precursors in the partial synthesis of various hormones such as testosterone, progesterone, etc. Whereas the saponins are also found to increase the rate of absorption of many pharmacological active substances, specifically the cardiac glycosides. Many saponin containing drugs are used as expectorants. The examples are ipecac and licorice, as their contents of saponin stimulates the bronchial secretion and also activate the ciliary epithelium of the bronchi. Although they are generally bitter, there are some exceptions, and the example is the triterpenoidal saponin glycoside glycyrrhizine, which is the main sweet principle of licorice. It is the calcium and potassium salts of glycyrrhizic acid, which in turn is the diglucouronic acid glycoside of glycyrrhetinic acid. Beside being a valuable flavoring and sweetening agent, licorice has demulcent, expectorant and antispasmodic action. All these activities are attributed to the saponin glycoside known as glycyrrhizine. Recently, glycyrrhizine was also shown to be effective in gastric ulcer treatment and is found to exert cortisone-like action in the rheumatic arthritis and other inflammatory diseases. Many saponins have strong spermicidal activities and even hemolytic activity. Monodesmosides are far more hemolytic than bisdesmosides. More than few saponins are even cytotoxic. For example, tubamoside 1 from Bulbostema paniculatum is cytotoxic in nature, while other may inhibit the tumor formation. Dear friends, I hope you have got a reasonable understanding about the saponin glycoside, their history, their occurrence, their physicochemical properties, their structural variations, the classification and even the identification test which we had used in order to identify them. All these have given a lot of difference in their pharmacological actions. If you have missed any of the module or content, you may log on to our website, that is www.cec.nic.in, to download videos, FAQs, LORs and other contents so that you can stay updated with this chapter. Now let's start with the next module, which is known as flavonoid glycosides. Flavonoids are the natural organic compounds with a phenyl benzopyrene ring containing 15 carbon atoms, which are arranged as C6, C3, C6 compounds, in which C6 moiety is a benzene ring, while the variation in the state oxidation of the connecting C3 moiety determines the properties as well as the class of such compounds. They belong to the polyphenol group and are present in free as well as glycosidic form, generally as O-glycoside or C-glycosides. Flavonoid consists of aromatic ring A, which is condensed to the heterocyclic ring C and is attached to a second aromatic ring B, as you can see on the screen. The structure is known as flavonoid diphenyl propane skeleton. Now let's see where they occur. They occur in most of the parts of the plants. These compounds generally occur in plants in the form of glycosides in different derivative form like flavin, flavanol, flavanone, isoflavone, chalcone and anthocyanins. Flavonoid glycosides contain sugar chain which is attached to one or more phenolic hydroxyl groups. The hydroxyl groups are generally found in position 5 and 7 in ring A, while ring B commonly carries hydroxyl or alkoxyl groups. Majorly, they are divided into two subclasses, anthocyanins and anthocyanthins. Anthocyanins are the glycosides of anthocyanidine and are the most important class of water-soluble plant pigments which are responsible for various beautiful colors such as purple, blue and red to the fruits, flowers and even leaves, while anthocyanthins are white or yellow or colorless compounds which includes flavanols, flavinols, isoflavones and even flavanones. Flavonoid found in various plants with their basic chemical structure as you can see on the screen. The first type of derivative is called as flavanol, which are present in the form of myrcetine, quercetin, camphorol. The basic difference in the structure is they have an hydroxyl compound at position 3 and a oxygen at position 4. The second type of flavonoid glycosides are flavanols, where they have only the substitution at C3 with a hydroxyl group. The examples are catechin and epicatechin found in the plants like green tea and pear. The third type we have flavones, where we have the oxygen group at C4 position. The various examples are epigenin, luteolin and ficetine. Then we have isoflavones such as genistein and diazine. Similarly, we also have flavanones like hesperidine, naringine and hesperidine, which are mainly found in the orange and grapefruits. Now let's see how do we identify the flavonoid glycosides. Flavonoid glycosides contain conjugated double bonds and thus they show the prominent absorption in visible and UV region between 470 to 560 nanometer. Their color helps their easy identification. Especially flavanol glycoside appears as pale yellow color in visible light while dark brown in color under UV light. Apart from all this, we also have several chemical tests which can help you in preliminary identification of flavonoid glycosides in the plants. Let's first start with the Shinoda test, where we can take the test solution. We have to add few ml of 95% ethanol, then 0.5 gram of magnesium turnings and a little of concentrated hydrochloric acid can be added drop-wise. If you can find pink, scarlet, crimson red or occasionally green to blue color, that means the flavonoids are present. It suggests the presence of flavanol, dihydroflavanol or xanthine derivatives in the sample. Coming to the next test, which is called as sulfuric acid test. Take sample and add sulfuric acid to the sample. A deep yellow color suggests the presence of flavones or flavanols, while the formation of deep orange color will indicate the presence of flavins, and reddish blue solution indicates the presence of chalcons and orons in the sample. Now let's come to the third test, which is known as alkaline reagent test. To the test solution, add few drop of sodium hydroxide solution. Intense yellow color is formed, which turns to colorless on addition of few drops of dilute acid. This shows the presence of flavonoids. Similarly, we also have zinc hydrochloride test, in which we have to take the test solution and add a mixture of zinc dust and concentrated HCl to it. It gives red color after few minutes. This deep red to magenta color shows the presence of flavonoids, while the light pink or no color may indicate the presence of flavonols. Another test which we can use to identify flavonoid is ammonia test, where we have to soak a filter paper or dip a filter paper in the alcoholic solution of a test drug and then expose to ammonia vapors. If you find the formation of yellow color spots on the filter paper, then it suggests the presence of flavonoids. So these are the various tests which we can use for identifying the flavonoid glycoside either in the extract or the drug sample which we have received for the preliminary identification. Now let's understand the pharmacological activities of the flavonoid glycoside. They are shown to exert different physiological activities such as cardiac stimulant activity, strengthening action on weak capillary blood vessels. Similarly, some highly hydroxylated flavons are found to act as diuretics and even as antioxidants to lipids. Flavonoid glycosides from ginkgo biloba exert anti-asthmatic and bronchodilator action. Similarly, rutin is used for treating capillary fragility and thus can be used to treat retinal hemorrhage. Let's now look upon to the module 5, which is going to discuss about the isothiocyanate glycosides. Isothiocyanate glycosides are the compounds of natural origin with isothiocyanate a glycon. That means it contains a sulfur and a nitrogen atom attached to the glycosides. They are also known as S-glycoside as they contain sulfur group and sometimes referred as glucosinolates compound because they usually contain beta D1 glucopyranosyl in the side chain of S-glycoside. They are mainly found in higher families such as Cruciferae, Caparidaceae and Racedaceae. The most famous drug containing isothiocyanate glycoside is black and white mustard, while glucotrapoline is found in Trapolium majus. Now let's look upon their structure. They are mainly glycosides of isothiocyanate a glycon which contain sulfur and nitrogen group as shown in the screen. They are mainly glucosides of isothiocyanate a glycon which contain sulfur and nitrogen groups as shown in the screen, where the R and the X groups varies in different drugs. They are found to be very unstable and break down by the enzyme myrosinase, which is also found to be present in the drug containing such type of glycosides. Now let's look upon to their pharmacological activities. They are usually found to exert an irritant or a disinfectant action, and they improve the blood circulation to organs when applied externally. On external application, they cause local irritation and they may cause inflammation of the skin too. Black mustard is the most important drug containing isothiocyanate glycosides. So let's look onto the black mustard. Black mustard are the dried ripe seeds of Brassica nigra and their varieties from the family Cruciferae, which contains the isothiocyanate glycoside known as cinegrine. They are majorly used as counter-irritant or rubefacient externally, while they are found to have emetic action when taken internally. Now let's have a little detail of cinegrine. Cinegrine is a glucosinolate which contains sulfur and nitrogen found in the seeds of black mustard, which is known as Brassica nigra and comes from the family Brassicaceae. It is also found in cabbage, broccoli, horseradish and Brussels sprouts. Chemically, cinegrine is known as allyl glucosinolate or 2-propenyl glucosinolate. Cinegrine is a precursor of allyl isothiocyanate. That means when a plant tissue containing cinegrine is crushed or damaged, an enzyme myrosinase is produced, which hydrolyzes cinegrine to an oil known as allyl isothiocyanate, and this allyl isothiocyanate has a pungent taste. Now let's look for its uses. It is used basically in our culinary, in food, and beside that, most glucosinolates are believed to stimulate the appetite as well as the digestive process. The oils are used as rubefacient, counter-irritant, and causes vomiting if consumed in significant quantity. Recent preliminary research has shown its antimicrobial, cytotoxic, and even anti-cancer activities. Now let's look for the module 6, which contains coumarin glycosides. Coumarin glycosides are found to be present in many plants, either in the free form or in the glycosidic form. They have derived their name from the plant tonka bean, which is known as coumaru in Caribbean island. It consists of naturally occurring compounds such as lactones, which are present with modification. So when a hydroxy group is attached, they are called as hydroxycoumarins. Examples are umbelliferone and esculatin. Similarly, if the methoxy group is substituted, then they are called as methoxycoumarin, and the example is scopolamine. While some which contains furano compound, then it will be called as furanocoumarins, and the example is soralane. These compounds show blue, blue-green or violet fluorescence under UV light. Now let's discuss about several coumarin derivatives. For example, esculatin, which contains a OH group at carbon 6 and 7. Similarly, fraxin, which contains a methyl substitution at 6, an alcohol substitution at 7, and a glucose substitution at C8. We also have herniarin, which contains methyl substitution at 7th position. We have scopolamine, where methyl substitution is found at 6th position and OH group is found at 7. We also have umbelliferone, where the OH group is substituted at 7th position. Coumarin glycosides are reported in more than 150 species, which belongs to 30 different families, mainly found in few families such as Leguminosae, Oleaceae, Rubiaceae, Solanaceae, to name a few. Some of the important plant drugs containing coumarin glycosides are Amivisnaga, tonka bean, Ami, and Soralia fruits. They are also found in few insects and beetles such as Cantharus, Vesictoria, and Mylabris. Let's see the few drugs which contains coumarin and furanocoumarin glycosides. The first one is tonka bean. These are the dried seeds of Dipteryx odorata coming from the family Leguminosae, and they are being utilized as flavoring agent. Then we have Visnaga. These are the ripe fruits of Amivisnaga coming from the family Umbelliferae. They are being used as smooth muscle relaxant and also as a coronary vasodilators. We also have fruits of Amimeges, which also belongs to the family Umbelliferae, and they are being used in the treatment of vitiligo. Not only this, we have another compound called as Sorolia, which are the dried fruits of the plant Sorolia coryliofolia from the family Leguminosae, and they are used in the treatment of leukoderma. Coumarin glycosides are mainly the derivatives of benza-alpha-pyrrole, whereas the furanocoumarins are formed by the fusion of furan ring to the coumarin at either 6,7 or 7,8 carbon atoms. Now let's look upon its pharmacological significance. They exert variety of physiological actions which differs with their forms. The major coumarin lactones was used as a flavoring agent, whereas the furanocoumarin derivatives found in Visnaga are used as smooth muscle relaxant and vasodilators. Some of the drugs containing this coumarin derivatives, such as Sorolia, is found to be used in the treatment of leukoderma, leprosy, psoriasis. Others found in insect and beetles are acting as a local irritant and rubefacient. Now let's look upon to the chemical test which is used for the identification of coumarin glycosides. Coumarin glycosides can be generally identified by their aromatic odor and fluorescence shown under the ultraviolet light. Remember, we said they give blue color, bluish green color or white fluorescence under UV light. The alcoholic extract, when made alkaline, shows blue, blue-green or violet fluorescence under UV light. Now let's look upon to the detailed procedure of the identification test used for coumarin glycoside. Place a small amount of sample in the test tube and cover the test tube with a filter paper, which is moistened with the dilute sodium hydroxide solution, and place the covered test tube on a water bath for several minutes. Then remove the paper and expose it under the ultraviolet light. The paper should show yellowish green fluorescence. If it shows yellowish green fluorescence, that means it shows the presence of coumarin glycoside in the test drug. Now we have the last module talking about the bitter glycosides. Bitters are the substances of natural origin whose alcoholic extract, if taken before meal, helps in improving the appetite. They are usually found to be bitter in taste but are safe for consumption. They are generally used as stomachic and digestive substances, while some of them are also used as sedative and tonic for stomach. Drugs containing bitter glycoside acts as bitters, stomachic, digestive and febrifuge. They are mainly found to occur in the families such as Compositae, Gentianaceae and Labiatae. Most common and famous drugs containing bitter glycosides are Chirata, Kalmake, Gentian, Picoryza and Quassia, to name a few. They are found to be present sometimes in entire herb or either in dried leaves, tender shoots, wood, roots and rhizomes of the plant. Now let's talk about the drugs which contains bitter glycoside. Gentian, which is a dried partially fermented rhizome and root of Gentian lutea coming from the family Gentianaceae, is used as a bitter stomachic and tonic. Similarly, we have Picoryza, which is the dried rhizomes of the plant Picoryza kuroa coming from the family Scrophulariaceae, which is not only used as a bitter tonic but is also a very important hepatoprotective drug. The next in the list is Quassia. Quassia consists of dried stem wood of Picrasma excelsa coming from the family Cimarubiaceae, and they are being used as bitter stomachic and even enthelmintic activity. And then we have the most important, known as Kalmake, which consists of the dried leaves and tender shoots of the plant Andrographis paniculata from the family Acanthaceae. These are not only used as bitter tonic and enthelmintic drug, but they are also very strong hepatoprotective agents. Bitter glycosides may not belong to the same class, but mainly they are the glycoside of monoterpene, diterpene or sesquiterpenoid iridoids with pyrene cyclopentane ring. Bitter glycosides are mostly used for their stimulant effect on gustatory nerves in the mouth, which further leads to an increase in gastric juice secretion and thus aids digestion. Dear friends, now you have got a fair idea about flavonoids, coumarin, bitter and isothiocyanate glycosides, with a special reference to cinegrine, their occurrence, chemistry and pharmacological significance, along with the chemical tests which are used for their identification. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, FAQs, LOR and other contents so that you can stay updated with this chapter. To conclude, let's have a reminder session. In the present discussion, we learned about the most important types of glycoside known as saponin glycosides. Saponin glycosides are known to exert structure-related diverse biological activities. Licorice, Dioscorea, ginseng, brahmi and shatavari are some of the important drugs with varied biological activities. Recent studies on saponin glycosides are focused on to their role in diabetes, cancer, contraceptives and so on. Some are investigated for their role as an adjuvant in vaccines. Similarly, there are few important lactones under coumarin glycoside such as psoralen, which has its effect in the leukoderma, leprosy and psoriasis. Not only this, but we have flavonoid glycosides which are also found to have various chemical groups and thus can be used in several ways. Today, we saw few more types of glycosides whose pharmacological activity greatly depends on their structural moiety of the erglycon molecule. With this, we came to an end of today's lecture. Hope you enjoyed our long learning journey with various glycosides having different medicinal uses. Don't forget to review all of them in your self-study. This is Payal Dande signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quizzes and even LORs. Our website is www.cec.nic.in. Till then, keep studying, goodbye and keep learning more with us.